The nervous system. There are 5 functions of the nervous system. The nervous system receives sensory input or sensory information from the external and internal environment. Next, through integration, all of the sensory information is brought together. Once all the sensory information is integrated or combined, the nervous system interprets what that sensory information means. When all of the sensory information is interpreted or understood, the nervous system can then initiate a motor response or action to that initial sensory input. These functions of the nervous system allow the nervous system to maintain a normal, stable, internal environment, or in other words, maintain homeostasis. The nervous system has 2 major divisions. The first division is the central nervous system. The second division is the peripheral nervous system. And the peripheral nervous system can be further subdivided into the sensory division and the motor division. And the motor division can be further subdivided into the somatic nervous system and the autonomic nervous system. And finally, the autonomic nervous system can be subdivided into the sympathetic division and the parasympathetic division. The main cell of the nervous system is the neuron. A neuron is a nerve cell that receives stimuli and transmits action potentials to other neurons or effector organs. There are several parts of a neuron, with each part serving a specific function for that neuron. The first part of the neuron is called the dendrite, and the dendrites are short extensions that receives information from other neurons or sensory receptors. The next part of the neuron is called the cell body. The cell body is the part of the neuron that contains the nucleus and other organelles typically found within a cell. Following the cell body is the axon hillock. The axon hillock is the location where an action potential is created. From the axon hillock, the next part of the neuron is called the axon. The axon is a long extension that conducts and transmits action potentials. Surrounding the axon is a covering called the myelin sheath. The myelin sheath serves as an insulator that prevents electrical current flow across the cell membrane. And between the myelin sheaths are gaps where electrical current flows easily, called the nodes of Ranvier. And the last part of a neuron is the axon terminal. The axon terminal is the end of the axon that synapses with dendrites of adjacent neurons. One way that neurons can be classified is by their function. Functional classification of neurons is determined by the direction the action potential travels between the central nervous system and the peripheral nervous system. And there are 3 functional classifications of neurons. The first functional classification is the motor neuron. The second classification is a sensory neuron, and the third classification is an interneuron. A motor neuron, by definition, is a neuron that transmits an action potential from the central nervous system to the peripheral nervous system. And it is the neuron responsible for creating a motor response or action. A sensory neuron is a neuron that transmits its action potential from the peripheral nervous system to the central nervous system. And it is the neuron that receives and transmits sensory information. And the last functional classification of a neuron is the interneuron. The interneuron transmits electrical impulses between a sensory neuron and a motor neuron. And the last topic of discussion about the neuron are the types of cell membrane transports found on the axon. And there are 3 types of axon membrane transports. The first type of transport is called a voltage-gated ion channel. This is a passive transport. In other words, it allows things or substances to pass through it by the process of diffusion or osmosis. The second type of membrane transport is called a chemical-gated ion channel. This too is also a passive transport. And the third type of cell membrane transport is the pump. The pump is an active transport, which means that it requires energy to move substances or materials across the cell membrane. A voltage-gated channel only opens in response to the presence of a voltage, or an action potential. When an action potential arrives down the cell membrane, this channel opens, allowing substances or materials to pass right through it by the means of the process diffusion or osmosis. A chemical-gated ion channel opens in response to the presence of a chemical, in this case a neurotransmitter. So when a neurotransmitter binds to a receptor, the channel opens, allowing substances to diffuse across the membrane. And finally, the pump, which requires energy, will transport substances across their concentration gradients to their resting locations. In this next section of the nervous system, we're going to talk about membrane potentials and how electrical impulses are transmitted and transported throughout the nervous system. A membrane potential is a charge, and in order to measure the charge of an axon, we have to look at the outside charge of the axon in reference to the inside charge of the axon. At rest, when no ions are moving across the cell membrane, the inside of the axon is negatively charged, while the outside of the axon is positively charged. Because there is a difference in charge across the cell membrane, the neuron is said to be polarized. At rest, the axon has a charge across the membrane called a resting membrane potential. The resting membrane potential of an axon is roughly around -70 millivolts. There are several factors that contribute to the resting membrane potential. The first factor is that there is a high concentration of sodium ions outside of the axon and a low concentration of sodium ions inside the axon. The second factor is that there is a high concentration of potassium ions inside the neuron and a low concentration of potassium ions outside the neuron. And finally, the last set of factors that contribute to the resting membrane potential are the closing of the voltage-gated sodium ion channels and the closing of the voltage-gated potassium ion channels. These channels being closed prevents sodium and potassium ions from flowing into or out of the axon. The movement of sodium and potassium ions across the cell membrane creates another charge called an action potential. In order to create an action potential, a minimal amount of stimulus called threshold is needed. Threshold has an approximate numerical value of roughly -55 millivolts. An action potential follows the all or none principle. In other words, if threshold is reached, an action potential is created. If threshold is not reached, no action potential is created. There are 4 phases of an action potential. The first phase is called resting state. The second phase is called depolarization. The third phase is called repolarization. And the fourth phase is called hyperpolarization, or the undershoot. When looking at a graph of an action potential, you also have to take a look at what is happening at the neuron, specifically what is happening at the level of the axon. During the first phase, called resting state, you can see that the sodium and potassium ion channels are closed. What does that equate to on the graph? Well, it creates a flat line at roughly around -70 millivolts, which we call resting membrane potential. During the second phase of an action potential, called depolarization, you can see that voltage-gated sodium ion channels are open, allowing sodium ions to diffuse into the axon, while voltage-gated potassium ion channels are closed, preventing potassium ions from leaving the axon. What does this equate to on the graph? Well, since you are moving positive charged ions into the axon, you get a positive ascension on the graph. In the next phase of an action potential, called repolarization, you can see that voltage-gated sodium ion channels are now closed, preventing sodium ions from entering the cell, while voltage-gated potassium ion channels are now open, allowing potassium ions to leave the axon. What does this equate to on a graph? Well, now you are removing positive charges outside the axon, which means the axon, in essence, the inside of the axon is becoming more negative, which is the descension part of this graph. And finally, the last phase of an action potential is called the undershoot. And in the undershoot, you'll notice that it has 2 parts. One part is where the graph continues to decrease, and then another part where the graph begins to increase, returning back to resting state. So the first part of the undershoot is due to, again, voltage-gated sodium ion channels remaining closed, but voltage-gated potassium ion channels are remaining open, allowing more positive potassium ions to flow out of the axon. The second part of the undershoot is due to the action of the sodium-potassium pump returning sodium ions and potassium ions back to their resting concentrations and resting locations, which is why we return back to resting state. The next type of potential that we will discuss is the graded potential. A graded potential is a small electrical impulse caused by a stimulus that is created at the postsynaptic membrane of a synapse. So looking at this illustration between two neurons, a synapse between two neurons, you have E1, which is the presynaptic neuron, and you have this yellow neuron, which is the postsynaptic neuron. When E1 sends an electrical impulse to the postsynaptic neuron, what's registered on the graph is this tiny little hump. This tiny little hump is the graded potential. So again, looking at the same illustration with graph from the previous slide, you can see that the graded potential created never reaches this thing called threshold. So remember, threshold is the minimal amount of stimulus needed to create an action potential. And it just so happens that this graded potential is not strong enough to reach threshold. Therefore, no action potential is created. So how do we get graded potentials to reach threshold so that we can create an action potential? And the answer is through summation. Summation of graded potentials can create an action potential. So in essence, you are basically adding graded potentials one on top of another until you reach threshold so that you can create an action potential. So looking at this second illustration here in the middle, E1, the presynaptic neuron, is sending an impulse, and it's continually sending an impulse, like rapid fire style, to the yellow postsynaptic neuron. And by sending this impulse rapidly, the graded potentials are never allowed to rest, and what happens is they actually summate or add upon one another until those graded potentials reach threshold and an action potential is created. This type of summation is called temporal summation. In the next illustration in graph, you see that now there are two presynaptic neurons, an E1 and E2. So when E1 and E2 are sending their action potentials are actually added upon one another, right, to create a larger stimulus, and this time that larger stimulus reaches threshold, again, creating an action potential. This type of summation is called spatial summation. Next, we're going to talk about what happens when two or more neurons meet. The junction between two or more neurons, or an effector organ, is called a synapse. And there are three parts to the synapse. The first part is the presynaptic membrane, also known as the axon terminal. The second part is called the postsynaptic membrane, or the dendrites or cell body of an adjacent neuron. And the third part is not pictured here, but it's found between the presynaptic membrane and the postsynaptic membrane. And this third part is called the synaptic cleft. So in order for an electrical impulse or an action potential to travel from one neuron to another neuron, a cascade of events takes place at the synapse. Assuming that an action potential is already created, how do we get this action potential over to this adjacent neuron? Before we do that, let's familiarize ourselves with the certain parts. So this part right here, this is the presynaptic neuron or presynaptic membrane. So this would be the axon terminal of one neuron. This is the postsynaptic membrane or postsynaptic neuron of an adjacent neuron. So this would be either the cell body or the dendrite of that adjacent neuron. And in between the presynaptic membrane and the postsynaptic membrane is this space called the synaptic cleft. So here are the events that takes place in order for an action potential to be passed on to an adjacent neuron. First, this action potential stimulates the opening of voltage-gated calcium ion channels, allowing calcium ions to diffuse into the presynaptic neuron. The diffusion of calcium ions stimulates synaptic vesicles to fuse to the wall of the presynaptic membrane. When the synaptic vesicles fuse to the presynaptic membrane, they release their neurotransmitter into the synaptic cleft. That neurotransmitter will then bind to a chemical-gated sodium ion channel on the dendrite of the adjacent neuron. So what happens is when this chemical gated sodium ion channel is open, sodium ions are allowed to diffuse into or across the dendrite's membrane. It's the diffusion of sodium ions across this membrane which generates a graded potential. So let's take a look at the overall picture of how an action potential from one neuron is passed to another neuron, or in essence, how do neurons communicate? So what we have is an action potential traveling down the axon of a presynaptic neuron. And once that action potential reaches the synapse, that electrical impulse is converted into a chemical messenger. And that chemical messenger stimulates the creation of graded potentials. And depending on the frequency of the stimulus or the strength of the stimulus, those graded potentials could possibly reach threshold. And if threshold is reached, a new action potential is created at the axon hillock of the adjacent neuron, and the electrical impulse will continue down that axon. If we look at the next picture, you can see that a stimulus causes a change in sensory receptors. And this change in sensory receptors generates an action potential, which will travel down an axon. And again, at the synapse, that action potential will be converted into a chemical signal or a chemical messenger. And that chemical messenger will help create a graded potential in the adjacent neuron. And if the summation of graded potentials reaches threshold, then a new action potential will be created, and that action potential will continue on down that axon.